I'm Alec, and today we will be talking about cellular respiration. Cellular respiration is cut into three stages: glycolysis, Krebs cycle, and the electron transport chain, also known as ETC. The general formula for cellular respiration is glucose plus 6 oxygen converts to 6 carbon dioxide plus 6 water plus ATP. Now, to start, we will perform glycolysis. We have our 6 carbon glucose, 2 ATP, 4 ADP, and NAD+. To begin the reaction, ATP will break apart the glucose model into two 3-carbon pyruvate. The glucose will lose 2 hydrogen atoms. These hydrogen atoms will then reduce NAD to NADH. The breaking of glucose will provide enough energy to turn the 4 ADP into 4 ATP by adding a phosphate molecule. This is substrate level phosphorylation. Now the pyruvates must get into the mitochondria, so the Krebs cycle and ETC reactions can begin. To do this, they bond with coenzyme A to make acetyl-CoA. The acetyl-CoA can then diffuse into the mitochondria. Now that we are in the mitochondria, we have the outer, inner, and intermembranes, as well as the matrix. The folds of the inner membrane are cristae and provide more surface area for reactions. Now that we know the parts of the mitochondria, it is time for the Krebs cycle. For the sake of time, the rest of the video will be done using only one pyruvate. So, acetyl-CoA will begin the reaction by bonding with a 4-carbon molecule named oxaloacetate. The coenzyme A is then released to bring more pyruvates into the mitochondria. This reaction makes a 6-carbon molecule called citrate. Citrate will then react with NAD. This reaction releases CO2, which diffuses out of the cell, and reduced NADH. Now we have a 5-carbon molecule called alpha ketoglutarate. This will then react with NAD again. This reaction again releases CO2 and NADH. However, it also provides enough energy to phosphorylize ADP to ATP. Now we have a 4-carbon molecule called succinate. Succinate then reacts with NAD and FAD to make NADH and FADH2. The 4-carbon molecule left is oxaloacetate. Remember, these are the products from only one pyruvate. You must double this to get a total of both pyruvates. Now we have reached the electron transport chain. ETC takes place on the inner membrane. Along the inner membrane, there are embedded proteins and ATP synthase. NADH and FADH2 have electrons on them that are released near the inner membrane. These electrons bounce through the proteins in the inner membrane. This energy opens the proton channels, allowing the H+ ions to be pumped from the matrix to the intermembrane. This is known as chemiosmosis. Now there are more H+ ions in the intermembrane than there is in the matrix. This causes a proton gradient. To keep the gradient equal, H+ ions must be pumped back into the matrix. This is done through the use of ATP. An oxygen molecule is the last electron acceptor. By accepting electrons, H+ ions are attracted to the oxygen, creating water. Also, when H+ ions pass through ATP synthase, it provides enough energy to phosphorylize ADP to ATP. This is known as oxidative phosphorylation. By the end of ETC, 32 to 34 ATP molecules have been created. This is the end of